Good morning, friends. Myself, Richa. So, today I'm going to talk about DNA replication and its tabular form of enzymes which are responsible for it. So, basically, replication it is a process of DNA makes a copy of itself. So, it is a synthesis of daughter DNA. You can see here two daughter DNA molecules are produced from one parental strand. Okay, one old strand is there, that is blue one, and one new strand is produced, that is green one. So two daughter DNA molecules are produced. That means the DNA replication is semi-conservative, right? And it is proposed by Messelson and Stull experiment. Next, what is the rules of DNA replication? It always takes place in the S phase of cell cycle. It occurs once per cell cycle. Next, it starts from OD, that is origin of replication. Next, it takes place in 5' to 3' direction always. Next, it can be uni or bidirectional in nature. Okay. Now comes the enzymes which are responsible here. Here you can see the DNA gyrase, or you can say topoisomerase. So here you can see it stabilizes DNA helix as it is unwound by the helicase enzyme. So first of all, helicase is responsible for unwinding of the DNA helix, or you can say unzipping by breaking the hydrogen bonds. Next comes the topoisomerase. This enzyme also it is of two types, 1 and 2. So basically it relieves topological stress in the helical DNA structure, right? So it is of one topoisomerase 1 and 2. Okay, so what does topoisomerase 1 do? So topoisomerase 1 makes a nick in one strand of DNA molecule. Okay, and topoisomerase 2 makes a double-stranded break in the double helix. So topoisomerase 1 and 2. So these questions basically what is the difference between topoisomerase 1 and 2? So always remember type 1 topoisomerase makes a nick in one strand of a DNA molecule, while type 2 will make a double-stranded break in the double helix. Next, SSBP proteins, which holds the unzipped single-stranded sections of DNA apart during replication. Next, DNA poly third, which adds new DNA nucleotides in the 5' to 3' direction, right? Next, primase, which add primers of RNA nucleotides to the lagging strand as starting points for replication. Next comes DNA pol 1, which replaces RNA primers with DNA nucleotides, right? And DNA pol 2 also works, but it works mostly in repair function. Next, DNA ligase, which join Okazaki fragments on the lagging strand. This ligase seals the gaps, right, to create a continuous DNA strand. So these are all the enzymes which are responsible for DNA replication. In this, basically, the helicases are motor proteins, you can see here, that couple the hydrolysis of ATP to nucleic acid unwinding. And it is hexameric helicase. Okay, it is having a ring-shaped structure, ring-shaped. Next, primase, you can see this enzyme is active only in the presence of other proteins like a helicase, which creates a complex called as primosome. Primosome. When helicase and primase will get combined, it will form a complex called as primosome, right? This question is frequently asked. So in the CSIR net exam in part C, they will give you a tabular form and they will mismatch the question and the respective function of the enzymes, and you have to find the correct one. So this tabular form is very much important. It's frequently asked in CSIR NAT exam. So you can remember all these enzymes. It's part C question and it's frequently asked here. Next, DNA ligase. This is also very important. The mechanism of DNA ligase is to form two covalent phosphodiester bonds between 3' and OH of one nucleotide with the 5' phosphate end of another, that is the donor one. That is important here. And DNA ligase in E. coli as well as most prokaryotes uses energy gained by cleaving NAD to create the phosphodiester bond. So here phosphodiester bonds are important. Diester bond. So that's it. Thank you.